execute the function of preventing the gene expression largely. There are different mechanisms of preventing gene expression that I am not getting into. So miRNA are produced by our own set of genes, the endogenous, and their function is primarily to regulate gene expression during development and as well as in normal adult function. And siRNA defends our body from pathogens like virus, viral RNA or viral DNA, any foreign genetic material gets silenced by this pathway. The piRNA based silencing protects the genome of our germline. So when I say genome, I mean the complete set of genes present in one particular cell. So when you talk about germ cells, these are the cells that give rise to egg in females and sperm in the males. And their genome needs to be specially protected because the survival of the species, like the integrity of the genome in the next generation, depends on the integrity of the genes in the germ cells. So therefore, germ cells mutations or invasion by foreign genetic element is not tolerated. So they have a special protective mechanism. That's where the piRNA mediated gene silencing plays a role. So in this work, I'm going to show you some evidence where we have identified a protein that we believe is part of this in mediating silencing in the germline. Okay, so that's the summary of my talk. So we do all of this using this wonderful model organism called Caenorhabditis elegans. It's a free-living nematode that you usually find in temperate conditions in rotten vegetables and fruits. This is the head to tail. It's about 1 millimeter, so you need a dissecting microscope to work with. So it's not this big a worm. And what's highlighted here is the gonad. So this is where the mitotically proliferating germ cells, then as they differentiate and mature as gametes, they progress in this direction. These are the oocytes and these are the embryos. So you have two gonadal arms. From both sides you have oocytes coming, and then here you have the embryos. And I'm able to show all of this because the cuticle of the worm is transparent. You can see through it. And that helps in recovering mutant phenotypes very readily. So in the interest of time, I won't get into the details. And it's very easy to get the gonad out. If you cut here with a needle, the gonad will come out like a nice long tube. Okay, so this looped structure is what is straightened here. The loop portion is here. So here you have the different stages of germ cell development spatially arranged. Sorry. So here you have the mitotically proliferating cells, here progressively in different stages of meiosis, and these are the oocytes and these are the embryos. Okay, so I'll skip these. Okay, so in a genetic screen, a former graduate student, Lenita Thomas, discovered an interesting phenotype for a mutant in a gene that that's called PLP1. I'll explain what it is in the next slide. Here the focus is on the mutant phenotype. So this is the gonad, the same structure as this, stained to visualize the chromatin of the germ cells. So each one is a nuclei here, what you see here. These are the oocytes. And in the PLP and mutant, the mutant here meaning it's not partial loss of function, it is complete loss of the protein coding part. So it's a null allele. When you grow at 20 degrees, that's the temperature at which we grow the worms in the lab generally, it's quite normal. It makes oocytes. Okay, it's like wild type. And when you grow at 25, the wild type worms are fine, but the PLP do not make oocytes. They only make sperm. Sometimes you do have mitotically proliferating cells where actually sperm should have been. And this is the phenotype she found, and this was interesting for us because we are always, as the introduction mentioned, we are interested in finding new genes involved in germ cell development. Okay, so we focused on this. So now what is this PLP1? PLP1 stands for per alpha like protein 1. What is per alpha? These proteins in other organisms originally shown to find bound to purine-rich single-stranded nucleic acids, DNA or RNA, and that's why they named it as Pura. And in C. elegans, they named it as Pura-like protein, so it is PLP1. So para-alpha was an intermediate name. Now people focus on using this. So this is present in all organisms from bacteria to human. Okay, and in vitro studies have shown and cell culture studies that it is found in RNA protein granule structures. So this is an alignment showing the relationship between the C. elegans PLP1 and the human per alpha. So there are conserved domains, and I'm not getting into that for today's part. So before us, people have done a knockout in mouse, 2 different groups, and they found problems with the brain development. So brain is, it's a section that's very enlarged, and this is a protein in the neurons that aggregates in the mutants, that's not in the wild type. And later people discovered through whole genome sequencing, some of the children born with neuronal abnormalities actually have mutations in different parts of this Pura protein, and these are the visible symptoms, and these children do not survive to adolescence. So it's a sporadic mutation. Before reproductive maturity, the child is dead. Okay, now increasingly more and more number of patients are being identified with this. Almost every month I read 1 or 2 papers describing a new mutation in human. Okay, so now getting back to our own story. So once Linitha found the phenotype, she decided to look at where it is expressed using antibody as well as a endogenously tagged fluorescence reporter, that is GFP. In both we found that it was present in the throughout the germ cells, okay, in the germ line, in all the germ cells we see it. And when she took a close look, it is found on granule-like structures that is around the nucleus in a perinuclear fashion. And when she used a marker for the nuclear envelope, she finds this is outside the nuclear envelope, it doesn't co-localizes with. And people do know RNA protein complexes present in this fashion already. And for example, one of them is PEGL1. We did a co-localization and that perfectly co-localizes, indicating that PLP1 is on those germ granules present in germ cells. And it's there in the embryo as well. In 2-cell embryo itself, the germline components are segregated into the one of the 2 cells. That's always the smaller one. So that is where we found the PLP1 granules, and some of them co-localize with the germ granule marker PEGL1. So then later a new student, Nihar, he looked at the other regions where we, Linitha had originally seen the expression. Like in the head region, it's found on the neurons as well as in the tails and in the ventral nerve cord as well. So it's present in germ cells as well as neurons in granule structures. That's consistent with what people have observed in cell culture studies where it is on RNA granules. Okay. So then Vishnupriya started to look at what does it do. And one of the things that we normally do is we focus on translation regulation. So therefore, we have reporters in our lab where the reporter expression is governed by the 3' untranslated region. So much of the translation regulation in the germ cell, germ line is via the 3' untranslated region. So we have a bunch of lines and she looked at them, and in one of them, what she ended up finding was in PLP1 mutant, this particular transgene, PAL1 UTR driven transgene is expressed in the oocytes, while it is not expressed in the wild type. So immediately we would think that PLP1 regulates translation via PAL1 UTR. But that's not the case, because wild type worms, when grown at 25, where germline gene silencing is defective, you see the expression there. So essentially what is happening is, for the silencing to happen at 20 degree, PLP1 seemed to play a role. So this is what we suspected, therefore we wanted to investigate and find whether that is true. So we looked at bunch of genes that are known to be silenced in the germline. Initially itself, remember I told you there is a special mechanism in the germline that protects against foreign genes. That mechanism does silence some of the endogenous genes as well in normal conditions, that is 20 degree. So some of the transgenes, for example this GAP gold one, is normally silenced in the wild type, and when we introduce into PLP1 mutant, it is expressed very nicely. And another transgene we tried, these are integrated single copy transgenes, and that is also expressed. So this again was consistent with our original idea that transgene silencing seemed to require PLP1. Then we looked at some extra chromosomal transgenes. So these are when you inject plasmid DNA with a promoter and a coding sequence and a reporter. These plasmids form a tandem array outside the chromosome and get transmitted in a non-Mendelian way. These will express nicely in the somatic cells, but they have telltale marks of foreign genes, so they get silenced in the germline. So one of them is this LET858 GFP. So these are the gonadal cells, somatic cells. So these are the gonadal somatic cells. There you see nicely the GFP, but the germline GFP is completely off. Whereas in PLP1 mutant, in addition to those gonadal sheath cells, you have germ cells also expressing. So for integrated transgene, you need this for silencing. For extra chromosomal array also, you need PLP1 for silencing. So next we decided to look at whether the piRNA mediated, which the piRNA are the small RNAs that are in core to suppress the transgenes and the foreign genes in the germline, including transposons. So we wanted to test what happens to piRNA mediated silencing. So one group had published a piRNA sensor where they have the promoter that drives expression in the germline, and GFP is our reporter. This helps in visualizing because this takes GFP into the nucleus, and this UTR doesn't have any translational control activity. So in this, when you put a piRNA target, so this is the complementary sequence to a given piRNA. So in the wild type, that piRNA will bind here and suppress it, and when Vishnupriya did this, so this is example to show what to expect. Like this paper has shown in PRG and another gene involved in this pathway, the silencing doesn't happen. In the wild type, it is neatly silenced. So here piRNA based silencing mechanism is defective, that's why it is expressed, and that happens in our thing too. So this is one more example, another piRNA we tested, and in both we do see it in the germline. Ignore this, I forgot to tell you earlier, this is autofluorescence of the gut granules. So these are the germ cells. So next we wanted to look at whether it is required for initiation of the silencing or for the maintenance of the silencing. So for that, if you introduce an already silent transgene into PLP1 background, then if it is required for initiation, then it will remain silenced. If it is required for maintenance, then of course in the absence of it, then it should get turned on. So what we found is it doesn't get turned on. So this is a control wherein this mutant which is required for this protein is required for maintenance, and you do see robust expression. So this we repeated for all the three classes, and we found in all cases it's required only for initiation but not for maintenance phase. So this is where we stand. If I have a couple of more minutes. So then we wanted to get into the mechanisms, and Andy Fire and his postdoc Lamia helped us to sequence the small RNAs to see whether small RNA biogenesis itself was affected in PLP1. So they found that they are not affected. So this data is from them. And then we looked at all the mRNAs, like we did transcriptome profiling. And transcriptome profiling means essentially you sequence all the mRNA in the two different conditions, in this case wild type and mutant, and look at which mRNA is increased or decreased in your mutant in comparison to the control. We found two groups, collagen related and sperm related genes are significantly upregulated. In total, we found 780 mRNAs are upregulated, some 589 downregulated. So we are trying to tease apart what is required and why they are upregulated, etc. So this is validation of this whole genome sequencing result. So that I'll skip. So another evidence to support our conclusion that PLP1 is required for germline silencing is we looked at the genes upregulated in other pathway components like PRG1 that I mentioned earlier and MUT16 another one. So the upregulated groups largely overlap. So that's what these Venn diagrams tell you. And among them, predominant group is the sperm-related genes. 96% of the sperm-related genes upregulated in PLP1 mutant are upregulated in both PRG1 and MUT16 as well. So in summary, I have told you that PLP1 acts downstream of small RNA biogenesis because Andy and Lamia showed it's not required for that. And we have shown that it is for the initiation, not for the maintenance. And from the two introductory slides, I showed you about mouse and human data, and we kind of think that this is not specific to germline gene silencing. PLP1 probably plays a role wherever RNA protein granules regulate gene expression. So that's what we are trying to figure out currently in the lab. So with that, I'll thank the funding agencies and thank you all for your attention.
Okay, time for one quick question.
Yeah.
How good is the homology between MI, SI, and pi?
Oh, so only the size-wise they are similar, but sequence-wise no, because these are each one have their own specific targets. So piRNA are tens of thousands of them. They pretty much cover the entire genome. And you have hundreds of miRNAs, each one specific for a given mRNA, or a group of mRNA at least.
So piRNAs are the most diverse in terms of sequence.
Correct, correct.
We have time for another quick question.
Yeah. Well, ongoing experiments suggest so. Probably next time I'll tell you for sure. Yeah.
So are piRNAs expressed only in the gonads or?
Yes, piRNAs were originally discovered in germ cells, but I can't say certainly that it is not existing elsewhere. It's that people have not found yet.
All right, so let's thank Professor Subramaniam once again. So our next speaker is Dr. Navin Bist. This is a talk of a completely different flavor. Dr. Bist is a plant geneticist and serves as a senior scientist and a group leader in the BRIC National Institute of Plant Genome Research, New Delhi. His research focuses on plant secondary metabolism and nutritional genomics in Indian oilseed mustard. Dr. Bist is a recipient of various prestigious awards, including the DBT Tata Innovation Fellowship, the DBT S. Ramachandran National Biosciences Award, DBT Innovative Young Biotechnologies Award, the NACI Scopus Young Scientist Award, the INSA Young Scientist Award. He is also an elected fellow of all the three Indian Science Academies and the National Academy of Agricultural Sciences. He was elected a fellow of the Indian Academy of Sciences in 2023. Best.
Thank you very much, Dr. Basu. I hope I am audible. Yeah, so great thanks to the Academy also to confer this fellowship in 2023. So today I would be talking more about my research, which I've been doing at BRIC NIPGR, which is under one of the plant sciences institution under Biotechnology Research and Innovation Council. And my group has been working in this very crop, Indian oilseed mustard, Brassica juncea, which we know. This crop is quite important for Indian subcontinent for many reasons. I am going to talk more about it. And primarily we work more to understand how these specialized metabolites which are present in Brassicaceae family members are being synthesized and can be used for human values. This crop, I'll just a brief introduction about this crop. This is a very important oilseed crop after soybean. It is cultivated in almost about 1/3 of the Indian subcontinent region. Is mostly in the northern part of our country. And production wise, although it lags behind kind of soybean, but believe me, the oil quantity is almost much higher than soybean, what we get as soybean. And this is a very important oilseed crop in terms of edible oil, and also the oil cake which is left after oil extraction is a very good, important for livestock and poultry feed. Unfortunately, our country right now is importing almost about 2/3 of our edible oil through imports, which is a huge burden in our economy. And to meet this task, Indian oilseed mustard, which is almost about 40% of oil is there, it need to contribute about 20 million metric ton of oilseed production by 2050 or so. Now, there are major areas of improvement which need to be done to achieve this goal. We need to increase the productivity. Productivity, basically production per acreage. We can go either through pure line, but it has some limitation that one cannot achieve to a certain level after certain levels. Heterosis breeding, that is basically mostly based on hybrid seed production, is another way to achieve this. And there are some good examples in Indian oilseed mustard using cytoplasmic meal elasticity system as well as using GMS-based or genetic meal elasticity system. Yes, this crop is also infected by or infested by a lot of pathogen and pest. Sclerotinia, in the last 1 decade or so, Sclerotinia stem rot is one of the big problem for this crop, and also Aurobenkia, a root parasite, is coming in a very big way to kill this crop and to damage this crop under field cultivation. My group has been working mostly on improving the quality, oil quality and oil cake quality of this crop, mostly on quality improvement, and I'll be talking about glucosinolate as one of the trait. What are these glucosinolates? So these are the specialized metabolites which are present only in the Brassicaceae family member. All crops like Brassica juncea oilseed, which is a oilseed crop, not only this, mustard family crops like cabbage, cauliflower, all these crops contain these secondary metabolites or specialized metabolites, and these are synthesized from amino acids. So precursor, based on the precursor amino acid, they are named as aliphatic glucosinolates, especially if they are derived from methionine, leucine, or isoleucine, or they can be categorized as aromatic glucosinolates, like which if they are derived from methionine and tyrosine. So this is a basic structure of glucosinolate. If you can see here, there is a glucose moiety linked to a sulfonated unit, and this R chain is the one which is contributed by the amino acid. Now, in a single crop, you can have almost about 30 different kind of glucosinolates ranging from these aliphatic or aromatic amino acids. These glucosinolates per se are not so active, but as soon as there is any pathogen attack or any wounding or any crushing of these crops, you found that these glucosinolates are degraded by myrocinases, which are a class of beta-thioglucosidase enzymes, which cleaves these glucosinolates into isothiocyanates form. And these isothiocyanates are the one which are very important for plant pathogen interaction, because they are the one which are toxic to pest and pathogen. So plant have retained them for their usefulness in terms of pathogen or on terms of plant defense. These glucosinolates and their hydrolysis products, especially these isothiocyanates, have various properties, like you can have positive effects, especially some of the glucosinolates which are present in broccoli. Glucoraphanin, they are known to be anti-carcinogenic. Also, they have a lot of chemoprotective properties in terms of when we consume these Brassicaceae family member crops. On the negative side, some of the glucosinolates, especially which is present in oilseed crop like Brassica juncea, they are goitrogenic. You have large amount of progoitin which competes with iodine, and thereby the oil cake which is fed to animals, they make retardation in the animals. So they are goitrogenic. Also the pungency factor, they also reduce the taste of the oil cake to the animal, and as a result, the animal do not like this protein cake or oil cake. So when we screened a very good all Indian oilseed mustard cultivar, and we found that there is very high amount of these seed glucosinolates. As a result, the high amount of these glucosinolates, the oil cake is not very useful for these poultry and livestock, and they are goitrogenic to livestock also. Also, they have very biting taste. As a result, the livestock do not like eating these protein-rich oil cake. Globally, if you see, there is a desire to have canola quality oil which are low in these glucosinolate fractions. So all Indian cultivars have almost about more than 100 micromoles of these glucosinolates, but these canola quality oil cakes should have less than 30 micromoles. So this is the standard levels of international levels which is acceptable for any oilseed crop to be consumed as a good crop. However, as I said, these glucosides are also very important defense arsenal of mustard family. So how can we achieve? So in the last 2 decades or so, we have been testing different ways to breed low glucosinate in glucosinolates in case of mustard family members, and we have used conventional breeding approach, genetic engineering, as well as very recently we have been using genetic engineering approach. And I will try to convince you how genome editing has helped us in a very big way compared to these previous approaches where we have done a lot of challenges. With Professor Pintel and Professor Pradhan, long back, I started this work. I worked with their group, and we worked out, can we do this? Can we breed low glucosinate trait, which is present in one of the non-Indian cultivar, to Indian cultivars, which are high in glucosinate? Can we breed it? So you can see this is quite a big pedigree. You keep on crossing this crop, this particular variety to this Indian type, then generate a line which is low in glucosinate, kept on crossing this with Varuna recurrent genome, and every generation, almost 6 to 7 generations, you keep on crossing. The major problem with this crop, this trait, is that it's a quite complex trait. It's quantitative traits. It's been governed by almost about 6 to 7 loci, which we know at that time. So there are 6, 7 major loci which control this complex trait. As a result, you need to breed all these 6, 7 loci from this variety into this variety, which was a big task for us. Almost about 6 to 7 generations it took. But what we found, even after 6 to 7 generations, there was a big problem of linkage drag. So although you do a lot of conventional breeding through traditional breeding, we found there is a problem of linkage drag, that even after many 6 to 7 generations, only 75% of this Varuna genome we were able to have. Still, there are 20-25% of this Hira genome was coming into the advanced generation lines. So this was a big challenge for us, that how to remove this kind of negative linkage of seed glucosinolates and the seed quality traits and the yield traits, which is coming because of the linkage drag. So we thought, okay, let's try something using GM approach. We know that there is a transcription regulator. This is a pathway of glucosinate biosynthesis starting from methionine. You found that there are n number of genes. Luckily, we found there was a MYC28 transcription factor homolog in case of Brassica juncea, which regulate these pathway genes together. So this is a positive transcription factor which regulates so many genes. We did RNA-based silencing of this RNA MYP28 sequence, and what we found that yes, in a single generation, we can generate lines which are less than 30 micromoles. No linkage, no problem of linkage drive, nothing. And what we found, we took these lines for event selection trial under open field condition. We found that yes, these lines are behaving very nice in case of for all the seed quality traits, only except the glucosinate trait which was coming down. So no compromise on seed yield and seed quality parameter, but the amount of time it took us to do even these event selection trial under open field condition was too much. Almost 2 to 3 years it took us to get these event selection trial permission. And now we will be taking these lines for BRL level trials. But the kind of work it took us to take these to the field level condition, still we know that GM still have some lot of it's under regulatory issues. So, but in both of these approach, as I said, these cannulata quality lines which are reduced in glucosinolates in seed and in leaf also, they are expected to have, they are highly vulnerable to pest and pathogen, because what we are doing that we are reducing this defense compound throughout the plant. So what could be a better strategy? We wanted to have a solution to develop an ideal line which will have only low amount of glucosinate in seed and retaining high amount of glucosinate in leaves, because so that these crops are not vulnerable to pest and pathogens, especially under Indian subcontinents. So we screened a big population of Indian mustard. We found that in all the population, there is no cultivar which is having low amount of seed glucosinolates and having high amount of leaf glucosinolates. Everywhere you find most of these cultivars are having very, very high amount of seed glucosinolates. We mapped this trait using genome-wide association and transcriptome-wide association studies, and we found there are many candidate genes which are promising base for this pathway. And luckily, we found one transporter gene which is known to transport glucosinate from leaf to seed. What does this do? In glucosinate, what is known that glucosinates are synthesized in leaves and then they are transported to seeds. We thought, okay, let's try this mechanism of doing the transport engineering. How can we use transport engineering to breed a mustard line which can accumulate low amount of glucosinate only in seed, but not without compromising glucosinate within the leaf areas or any other tissue types. But what we found is Brassica juncea is a crop which is allotetraploid from two different genomes, A genome and the B genome, and there are 6 homologs of GTR1 and 6 homologs of GTR2. So quite a complex engineering we need to achieve for this crop. There are 12 glucosinate transporter homologs in this crop which need to be tackled. But before that, we found that most of these transporters in fact are expressed very well, but catalytically we have no idea whether they are active or not. And there are no assays for transport activity to check also how we can do it. So one of my PhD students started working on transport assays to work out how to do it. Luckily, we found a cotton cell line. Cotton is a plant where these transporters are not expressed because these secondary metabolites are not expressed. So we developed cotton-based cell lines and we expressed these transporter there. And what we found when we expressed these transporter, we found that all these transporter, all the 12 transporter which we observed, were catalytically very, very active. They can transport glucosinate, different kind of glucosinate to a very high affinity. So these transporters are catalytically very active and they are also expressed ubiquitously. So this became our task even more challenging. Two of my graduate student, PhD student again work on this, and we worked through editing approach. How can we now, our goal is to edit all the 12 transporter, all to do mutation in all the 12 transporter. Luckily, we found a conserved genome, exonic region especially in the second exon of the gene, which seems to be very much conserved, and we designed 3 guide RNA using multiplex genome editing approach. We transformed Brassica juncea and tried to in the mega variety of Indian mustard, and we developed lines which are editing. We found that these lines could do this multiplexing can do editing of most of these transporters. Almost up to 10 transporters are edited in the advanced generation. There is a biallelic mutations for same kind of mutation in advanced generation. So this is something significant because most of the glucosinate in crop breeding, most of the genes which has been edited so far are only few, and this is the first success stories where up to 10 genes in a single plant has been edited by CRISPR-Cas9 based genome editing. And what was the effect of this transport? We found that the low amount of in the seeds, the amount of glucosinates are now very, very low, less than 30 micromole of international standards in all the genome editing plants which we have generated. The leaf glucosinolates was not compromised at all. In fact, it was much higher because the glucosinate transport was no longer going to the seeds. So the leaf levels of these glucosinolates, these defense compounds are quite high in leaves. Also, very interesting fact was that for the first time we reported that these glucosinolates are transported from pod wall to the seeds, and pod wall also accumulate a good amount of these glucosinolates even in the edited lines. So this is something that, okay, it's a win-win situation that only the developing seeds are accumulating very low amount of glucosinolates, but throughout the plant, all across on all the tissue types of this plant, we have high amount of glucosinolates, which is in fact very good for Indian subcontinents where you have a lot of pest and pathogen problem. So the benefit of these transport assays is that the engineering is that we have developed ideal canola quality mustard with improved oil quality. You have low amount of glucosinolates good for oil quality as well as for oil cake quality. Improved seed meal quality for livestock and poultry feed, and also because of high amount of glucosinolates, these are more protected against pest and pathogen under field condition. And I will show you one data. We have through RCGM based guidelines, we have kind of taken these lines under open field conditions, done station trial, as well as these lines have also undergone ICR advanced varietal trial. And I'll show you just one data to convince you that on the left hand side you can see on your right hand side you can see this. There is a conventionally bred canola quality lines which is heavily infected by termite attack during the maturity. But all our genome editing lines which are low in seed and high amount of leaf glucosinolates, they are intact, they are growing well, and they are forming very good seed set under open field condition in multi-location trials also. So this is something very significant that by engineering glucosinate, tissue specific engineering of glucosinate, we can develop edited lines which are very useful for nutrition as well as for defense traits. Very recently, what we have done, not only we have reduced glucosinate from the seeds, another very important point, as I mentioned, that glucoraphanin is one of the nutritional glucosinate which is present in broccoli, and broccoli is consumed because of this particular compound, natural compound, which is known to be anti-carcinogenic. So we thought, okay, glucoraphanin, although glucoraphanin is very high in broccoli, but broccoli is something not very not very well cultivated in Indian subcontinent. But this Indian mustard, which is also used as a saag and oil cake, oil and oil stuff, can we try to engineer this particular glucosinolates in Indian oilseed mustard? We know there is one candidate genes, which is AOP2 genes, which convert this glucoraphanin into these anti-nutritional compound, glucoraphanin and progrotin, which are anti-nutritional. So by editing, what we have done, there are 5 homologs of AOP2 in Juncea, and what we have done by editing 5 of these homologs, we have generated lines which are having very high amount of these green colored, if you can see, glucoraphanin, which are very, very high. Almost about 75 micromoles of glucoraphanin is now present in Indian oilseed mustard. which is the highest reported to date in any of the Brassica crops. So the benefit is that now this Brassica juncea can be used as new generation of superfruit, which can have chemopreventive benefits, and also it will have a lot of interstitial production of glucoraphanin and its hydrolysis product, which is sulforaphan. Regarding one last slide to show that yes, these glucosinolates can also be engineered for the defense properties. We know that in aliphatic glucosinolates there are different classes of glucosinolates, one which contain 3 carbon chain and another which contain 4 carbon chains. So these are 2 predominant class of glucosinolates in case of Brassica juncea. My student found that Bhanu found that when the C3 glucosinolates, particularly if you do some plate-based assays, these glucosinolates can take care of a lot of pathogen effects. The growth of the pathogen Sclerotinia specifically is reduced to a very significant level, even at 1 micromole. But unfortunately, all Indian cultivars contain very high amount of these longer chain glucosinolate 4C. So can we try to engineer this? And we have done some very interesting work in 2019, where we've shown that there is one class of genes, protein methyl alkyl malate synthase. It converts short chain glucosinolates into long chain glucosinolates. So right now what we are trying to do is that there are 4 homologs of these methyl alkyl malate synthase gene. We are trying to achieve base editing of these residues which catalyze shorter chain and longer chain glucosinolate substrate so as to form only those glucosinates which can do only those MAM to homologs kind of stuff, where we can have conversion only for smaller chain glucosinates. So that we can develop lines, Indian mustard lines, which can have accumulate more of these C3 glucosinate in our Indian cultivars. So this is something which we are trying to work using genome editing for by precise base editing. We wanted to do this engineering for higher defense attributes also. To sum up, we have done some contributed something on glucosinate in biology in Indian oilseed mustard. In aliphatic glucosinolates, there are many areas which we have investigated. In fact, in all these areas, right now we are working on another compound, indole glucosinate or aromatic glucosinate. We have some interesting lead, and hopefully in next whenever I'll talk, I will try to express some of my expression of these stories which are coming up in a very good way. Thank you very much, and I acknowledge here all my current and former director of BRIC and IPGR, lab members. I presented work of a few of my lab members, and many of them still there. My collaborators, Professor Deepak Mental, who has nominated me for this fellowship, Professor Pradhan, Professor Dr. Jagreet Kaur from University of Delhi South Campus, and many other collaborators, and funding from all different funding bodies. Thank you very much.
Thank you, Dr. Bis. We have time for questions.
Good afternoon. I just want to ask, are there certain soil conditions that are needed to grow this crop, or are the soil present in the northern India, or any cultivable land is okay to grow this?
So this crop is well adapted to northern part of our country.
So the soil present is the soil okay, or does it need some specific conditions?
So this crop is quite very interesting because it doesn't require too much of I'll say nutrient stuff. It is a kind of, it can be used in low tillage condition also. But yes, sulfur is one of the compound which is depend heavily on that compound.
Another question I have another question. So since it's an empty phyloid genome,
Yes.
So have you noticed any dominance in the subgenome in any type one of the types?
Yes. So this is always true with us because we have done a lot of subgenomic dominance studies in all our gene families. We found there are almost about 4 to 6 homologs, especially GTR also. We found there are 4 homologs of GTR1 and 2 homologs of GTR, 4 homologs of GTR2 and 2 homologs of GTR1, which are always dominant. We have some data. I'm not presenting it here, but yes, there is dominance. Yes.
Another one, sorry.
Yes.
I just want to ask, when you is there when there is an accumulation of GSL in the vegetative tissues, have you noticed cell toxicity, or does it alter disease resistance or something?
So yes, disease resistance, I've shown you data on the disease resistance. They are very much tolerant against many pathogen and pest under open field condition. No problem on any of the yield parameters. These lines have already been tested under open field condition in our station trial. Also, ICR is doing independent trials in 16 different locations. And couple of days back, I got news that one of our lines has been going to the advanced varietal trial, third year also. Yes, thank you.
So I'm not from plant sciences, but when you do these field trials,
Do you also harvest the seed and see whether the taste is acceptable or the taste
So,
changes?
We do not go up to taste levels because right now the permission for consumption of these lines are not there. But yes, we do all the nutritional tests. We do glucoside analysis. Also, we do many different seed.
Nutrition is okay, but the question is, will people accept it? Does it taste?
Oh yes,
good?
yes. Sure. Yes, because canola quality is still acceptable in India, and then we do expect that these quality lines will also be accepted. In fact, these lines are very good because globally there are no lines which are accumulating only low amount of seed glucoside and containing throughout all other plant parts. Yes.
So Naveen, very nice talk. Just was curious to know what type of mechanism GTRs follow, whether it's a uniport or a symport.
Oh, so this is a kind of this is an importer which transport glucosinate from the apoplast to the within the cells, and we have done some these transporter assays to show that these importers can import these glucosinate substrate within the cells, and that was the beauty of those transport assays also.
And there is no effect of pH or
Oh yes, they are
ion
pH
strength.
dependent. They are pH dependent. They are generally, the requirement is about 5.8 is the optimal requirement, and we have done those assays also.
Sure.
In our
Thanks.
plant method paper. We have shown that. Yes.
Hello, sir.
Yeah.
Sir, brassica is a crop involved in phytoremediation at times. Like it is a accumulant. So, is that property affected by gene modification? Is it?
We have not tested up to those
Okay,
levels,
sir.
but I'm sure these are very precise editing, and these transporters are doing job of only transporting
Okay,
glucosamides.
sir.
That much we know about it. Yeah.
No, sir. CRISPR also has some off-target
Oh
effects.
yes.
sir.
So this is very valid question. We have generated many lines, and I didn't see any problem with any of the other stuff. Right now, in fact, we are doing some deep sequencing of these lines to see if there is any off target, and I'll get
Okay,
back to
sir.
you.
Thank you, sir. Thank you.
All right, so if there are no more questions, let's end the speaker. So next speaker is Dr. Munia Ganguly. Dr. Ganguly is a chief scientist at the CSIR IGIB, the Institute of Genomics and Integrated Biology in New Delhi, and she is also a professor in the Academy of Science and Innovative Research. Dr. Ganguly has got her master's degree from Jadavpur University and her PhD from the Indian Institute of Science. She had done her postdoctoral research at the Phil's University of Pittsburgh for 2 decades. Her lab looks into the development of nanoparticles for gene delivery and application of nanotechnological tools. She has been awarded this fellowship in 2024. Yeah.
Thank you, Anulabu. I hope I'm audible.
Yes.
Okay. So, good afternoon, everybody. Deeply thankful for this opportunity to speak here. Today, I'll be talking about how to build targeted non-viral gene delivery systems. Why do we work on this? Because gene delivery systems are involved in what is called gene therapy. Gene therapy can come in various forms. It could be introducing a healthy version of a defective gene. Say, for example, there's a mutation in a gene that causes a disease. You can exogenously supply the healthy version from outside. It could also mean gene silencing, where you suppress the action of a defective gene. Say a gene has gone rogue and you want to suppress the expression. So can also be done by exogenously supplying a different form of nucleic acid. And then gene editing you already heard about, that's permanently correcting a defective gene. So to do all this, there are 3 things that one needs to sort of worry about. One is, of course, the gene target. You need to know which gene one can correct, and all diseases need not or cannot be treated with gene therapy. Second is the payload. Depending upon what change you want to make, It could be large DNA or RNA molecules that you want to send. It could be small RNA. It could be the CRISPR complex for editing. And the third thing is the delivery vector. So because nucleic acids are large negatively charged molecules, they don't get into the cells very easily. So you need what are called vectors to encapsulate them and stabilize them and send it to the cells. So viruses are the natural choice because viruses can get inside us very easily. So you can use them as carriers. But non-viral chemical methods are also gaining a lot of momentum these days for reasons that I'll be explaining in the coming slides. So while there is a lot of research in the academic space that's going on both viral and non-viral vectors, and we have come quite far in terms of development of viral vectors. If you look at the clinical trial space, of the 4,000 odd clinical trials that have happened, there are only about 40 odd products that are there in the market, and all of them, nearly all of them, use viral vectors for transport. And these therapies are extremely expensive. So the reason for this is that viruses, as I said, that they're extremely efficient system, but and that's the reason why most of the trials and most of the products are based on viral vectors. But they do have certain limitations. Cost is something that I mentioned. The scalability could also be a challenge. In most of the recent viral vectors that are being used, the non-integrating ones, the payload that you can send, the size of the nucleic acids that you can send is often not very large. And you have to make very complicated designs in order to increase the payload capacity. And there could be immunogenic reactions as well. So that's one of the reasons why, that's many of the reasons why people are trying to switch to non-viral vectors. However, in spite of very long, many years of research, we still don't have very good efficient non-viral vectors for gene delivery or gene therapy applications. There are only a very handful success stories in the clinic. There are plenty in the preclinical stages and even more in the academic space. So in order to design an efficient non-viral gene delivery vector, one needs to understand the barriers that this vector will face during its journey from, say, the needle to the nucleus. Okay, so these agents are usually positively charged, so they electrostatically interact with the nucleic acid and they form what are called nanocomplexes. And these nanocomplexes are, one usually overcharges them, as in use more of the agent to make it positively charged so that the colloidal stability is maintained. And when you send it through the systemic circulation, most of them, if the size is very small, they are cleared out by the kidneys. They can go and accumulate in large amounts in the liver. And if it manages to go through the bloodstream, they can get engulfed by the macrophages, they can get coated by the serum proteins, and all of these are barriers in the particle reaching its destination. And even when it comes out from the circulation, goes to the cell of its interest, the cellular membrane has to be, the barrier of the cellular membrane has to be overcome. And then there are endosomes that can, inside the cell, that can trap these particles and degrade them. So it's a very, very long journey, and the carrier design has to account for overcoming these barriers. So each carrier design sort of takes into account how one can counter these barriers. And even when you are, sort of not injecting something, sending it locally, topically perhaps, those organs have their own set of barriers, and this is something that I'm going to be talking about. So the main non-virals that have been researched extensively are lipid-based systems as well as polymer-based systems. And the main purpose is, again, how to overcome the barriers. And in the process, the lipid-based systems, for example, have evolved from very small liposomes to multi-component, 4-component, 5-component lipid nanoparticles. And, people are playing around with the head group chemistry, the linker structure, tail chemistry, so on and so forth. Same with the polymers as well, and then making hybrid systems and all that. So when we started working in this area, we wanted to look at systems that are more simple, maybe one component that can overcome all the barriers. So we started with peptides, and that's because they are easy to synthesize And the different amino acids have the different properties that are important to overcome these barriers. So initially we tried to only focus on overcoming the cellular barriers, and then as I'll show in my talk, how to make them suitable for certain organs, and where all we failed and had to change the system altogether. So after a lot of trial and error and some brute force, some little bit of intelligent thought process, we came up with this amphipathic peptide, which we called MGP9, and it's a 22-mer peptide. It has a lot of arginines. So these arginines are actually responsible for the interaction with the DNA because they are positively charged. We have histidines, which are ionizable groups, so they can actually help in the endosomal release. We put 2 cysteines at the end, which can form SS bonds, which stabilizes the particles outside, and inside the cell in the reducing environment, it can disassemble. That's also important. We made this secondary amphipathic, so that when it forms an alpha helix in presence of the membrane, one phase of the helix is all positive charge, one phase is all hydrophobic, and that we felt would be helpful in both the condensation and making the particles, and also to cross the cell membrane. And indeed, we could make monodispersed nanoparticles less than 100 nanometers in size, and with very good transfection efficiency across a large number of cell lines with very low toxicity. So we could optimize a carrier, a single component carrier, which is able to overcome the cellular hindrances of delivery. Now we wanted to sort of try to see if this can be applied in sending nucleic acids to different organs. So we chose skin first because it's the largest exposed organ of the human body, and it's easy to topically apply in the skin. But again, as I mentioned, that there are local barriers. So the skin has something called the stratum corneum on top, which is terminally differentiated keratinocytes that are embedded in a lipid matrix. So if you have to send something into the skin, you have to either go through the lipid matrix, or you have to go transcellularly. And while there are many methods that do either one of them, they could be invasive. The physical methods could be somewhat invasive. The chemical methods can be kind of abrasive in the sense that they can disturb the lipid matrix to an extent that the integrity of the skin is lost. So that's not an ideal situation. So what we thought is that because our carrier has, it's amphipathic and it has these hydrophobic residues in the alpha helix, it will form one phase which is hydrophobic. Maybe we'll be able to directly use this to deliver nucleic acid to the skin. So we took this peptide with a reporter plasmid DNA, and in all the data that I'm going to be showing, they're all reporter DNA unless I mention otherwise. And we made the complexes. We saw that, they not only enter the skin cells, they enter the skin tissue up to the lower layer of the epidermis. In the animal model, the SKH1 mice, which is a hairless mouse model, so it resembles the human skin in the sense that the follicular density is low. In normal mouse, the follicular density is very high. So obviously no follicular pathway can be operative here. We even formulated it with a chemical enhancer called silicone oil. And the best part was that there was no disruption in the skin structure on topical administration. We took what is called a Franz diffusion apparatus, put the skin tissue in between the donor and acceptor chambers, and we gave this formulation along with the skin impermeable dye, and we saw that the dye does not penetrate into the acceptor chamber, unlike with some commercial agents. So this was very nice, and we also worked out the mechanism by which it gets in. I am not going to the details. The next thing that we tried was that, okay, we could do it in skin. Can we do it also in another organ where we can locally apply in lungs, where we can give it, say, intranasally, or in case of a mouse model, you can give it intratracheally. can we use the same complex directly? So here we figured out that that may not be possible, because in case of lungs, if you have to reach the lung epithelial cells, there is in disease conditions, there's something called mucus that sits, and the mucus is made of mucin biopolymers. These are negatively charged. They're also very cross-linked, and that makes the mucus very viscous. And these positively charged particles are likely to get stuck in the mucus and not reach its intended cell type. So what we did is that we covered this surface with a molecule called chondroitin sulfate, which is a biopolymer, negatively charged biopolymer, to bring the charge down, and also it's a biocompatible molecule. And then we surface decorated it with mannitol. So mannitol is a water attracting molecule. So we thought that this will reduce the charge, put more influx of water, and perhaps it will be able to go through the mucus more easily. And this is what we observed, that yes, indeed, these particles, we prepared them, and with our collaborator, we did a series of assays in artificial mucus, where we could see that the modified particles, the green ones show the modified particles, they work better in terms of lowering the viscosity, movement in the artificial mucus, the diffusion, and also penetration. And even in a mouse, C57 mouse, when we intratracheally administered these particles, there was more accumulation of the mannitol modified particles as compared to the unmodified ones. So this was also very nice. One thing I wanted you to take note is that, okay, the charge does come down. We measured the surface charge, that does come down with the modification as expected. The size, however, goes up. That goes to about 400 nanometers, and I want you to take note of this for the subsequent studies. So the point that I'm trying to make here is that even when you have an optimized system for cellular delivery, even if it works in a certain organ, it's not necessary that the same system can work in a different organ. Depending upon what the barriers it will face, you have to make chemical modifications. So both these cases, however, there was no blood involved, right? So there is no passage. It doesn't have to go through the blood anymore. So the next challenge that we picked up was whether we can send these particles to the brain. So here, of course, you have to send it through IV. And as you all know that it is very difficult to send something to the brain because of what is called the blood-brain barrier. So you have these capillaries that are surrounded by the brain endothelial cells, and they also have these tight junctions. And then you have the astrocytes, pericytes, and the brain parenchyma. That is where you want to send the particles. So if you have to cross the BBB, you have to either disturb the tight junctions, or you have to make the molecules transcytose through the brain endothelial cells. And there are many approaches that people have taken in the literature. Each one comes with its own set of issues. So while we were researching, while we were figuring out whether our MGP9 complex can be sent for brain delivery, we came across a series of studies where people have shown that there are these small invaginations called cavuli, which normally are not present so much in the brain endothelial cells, but in presence of in a diseased condition or in presence of certain molecules, these cavuli, which are lined with a protein called caveolin 1, they open up in the brain, and people are using this as a strategy for delivering small molecule drugs into the brain. And we also came across a study that mannitol is a molecule that does also induces cavuli formation in the brain. So we were very excited that, okay, we already have a mannitol modified particle, so we can directly use this. But the problem was that the size of this cavuli is only about 100 nanometers, and as I mentioned earlier, that the particles that we have prepared are 400 nanometer in size, so no way they'll be able to go through this process. So I'll just take 2 more minutes. So we now realize that we have to perhaps change the carrier as well. We cannot do surface decoration. We have to introduce the ligand in the carrier itself. So we chose this type of polymer called polybeta amino esters. These are basically diacrylates and amines joined together in various combinations, so you can generate libraries of molecules. And they are off late, these molecules have been there for like 15, 20 odd years, but Of late, there's a lot of interest in these molecules because people are showing that depending on what monomer you use, you can actually send your cargo to certain intended organs. So maybe to liver or to lung or to spleen, depending upon the monomer chemistry. So people don't still understand very well how that happens, but that's something that is being discussed a lot. So Betsy, my student, what she did is that she made a mannitol diacrylate and she introduced that in the chain itself. So now when you complex, you have the mannitol on the surface, but it's not like a ligand. It's a part of your carrier itself. So no wonder the particle sizes did come down to 60 odd nanometers. We did all the other assays to sort of confirm the particle formation. And we did both in vitro, ex vivo and in vivo assays to see how it functions. And in the brain, both in the brain endothelial cells in culture as well as in neurons, there is good transfection efficiency. In ex vivo, when we create a BBB model ex vivo, there is good transport. And when we tried to do it in vivo, we thought that maybe the mannitol in the carrier may not be sufficient to induce cavuli formation. So we gave additionally a mannitol injection as well. And we also confirmed that this does not change the integrity of the tight junctions, and there is induction of caveolin 1. And after mannitol injection, when we send the particles, we do see strong signal in the brain, less signal in the liver. It does go to the neurons as well. It's not very clear from this picture, but you have to sort of take my word for it. We still don't know why it goes to the neurons in large amounts instead of the other cell types. We are still working on that. But again, the point here that I wanted to make is that sometimes, depending upon what barriers you have to overcome, you might have to also not just change the ligand or the surface decoration, but you might have to change the carrier itself. So going forward, what are we doing? We see that PBE has a very modular structure. So like in case of brain, we can now, make a series of PBAs that can be directed to different organs. And why a particular monomer combination or why a particular monomer ligand combination works, we are still working those out, but we do have several compositions that we can see are going into muscle, skin, brain specifically. So the mechanisms we still are working out. The other thing that we are trying to do is that, okay, all these I mentioned are only with reported DNA, just to show that the carrier works. Can we actually use them for a therapeutic application? So with a large number of collaborators, we have picked up 2 different diseases. One is TGM1 dependent congenital ichthyosis, which is a skin barrier disorder, and mutation in the TGM1 gene is what causes in not majority, but about 30-35% of the cases. And whether our MGP9 can be directly complexed with TGM1 and it can be sent as a, in the form of a plasmid, and whether we can see some functional application of that. The other one is called GNE myopathy, which is a muscle dystrophy, a kind of a muscle dystrophy. Again, mutations in the GNE gene cause that, and we are trying to use our PBA polymers to send the GNE gene in muscles in vitro and in vivo. Neither of these diseases have an animal model, so it's very hard to say that there's rescue function. But yeah, I'll not get into the data. There isn't enough time. But yes, so far we have initial experiments show that the overexpression is indeed working in the up to in vivo model. So we are now trying to see what kind of disease models we should look at. Maybe 3D skin structure in case of skin, and maybe some other model that we should maybe organize that we should look at as a disease model. I'll end here. I would like to thank all my present and past students. It's for them that I'm here. All my collaborators and their students, all the funding agencies who have been very gracious in funding the research throughout these years. Thank you very much.
Very, very interesting work and with clinical application. But the question I have is, skin has such a large surface area. If we apply it on the surface, will we be able to, in a disease such as ichthyosis, which involves a large part of the skin, will we be able to reach a level, or would it still be better to give it, can it still be given parenterally? Would that be better?
Yeah, it's a very good question. The good thing, I think, is that one can formulate and apply in large areas as well. That is one of the advantages. Instead of like, when you have to inject something, this is a topical administration. So you can actually cover very large areas theoretically. We haven't done that, but it's possible to do that. And since the system is also kind of non-invasive, safe.
Cost becomes a problem.
Non-virals are much cheaper than say if you want to do a gene therapy, if you have to use virals, and there is a product in the market with viral vector for not for ichthyosis but for what is that other not psoriasis, there is another rare skin disease, I'm just blanking out a little. So it's there in the market. It's with an HSV1 vector that people have used it. The cost compared to that is very minimal. So I would not worry that much about that. Whether it will work or not, and especially with such large surface areas, my hunch is that yes, it's worth a try. Yeah.
Only one quick question. This is very nice. Topical, I can understand, but when you're actually injecting it,
Yeah.
if you don't understand the process, how it targets a specific organ, it's not going to work, right? You need to understand that to be able to figure out where you want to deliver, the specificity of delivery.
Right. So that's what I'm trying to say. So the specificity of delivery, usually what people do is they use a receptor ligand chemistry. So the kind of ligands that I was talking about, those are not receptor ligands, but the idea is the same. You use ligands to surface decorate the particle, and those ligands will have receptors that they will target, and that's the standard mechanism of doing targeted delivery. Okay, it has a whole bunch of problems. So now the concept is also changing a little bit with lipid nanoparticles as well as with PBA polymers, that in the chemistry of your carrier itself, there are moieties that you can introduce which will help it take it to the organ of your interest, okay, and not send everything to the liver. Like when you inject it, major part of it goes to the liver. So how does that happen? It's still not known, but one of the theories is that the serum proteins that they accumulate on the surface of these nanoparticles, they sort of speciate them. So depending upon what chemistry your carrier has, those kind of serum proteins will come and bind. That will take it to either lung or to spleen or to liver, not exclusively, but with sufficient amounts that can give a therapeutic effect. So this is very much in the last 4 or 5 years in the lipid nanoparticle, for example, people have created what are called sort lipids. So they basically sort it to different organs. So this is a challenge, but there is also a lot happening, currently. And with the PBAs, it's again not worked out, but it is, I think, going in the similar direction as LNPs. So that's also something that we are trying to figure out.
Yeah. So, on the question of cost of viral vectors, I mean, we've all been injected, right, with the COVID virus. That's $5 a dose. It was.
Yeah, that's
not that high.
That's a vaccine.
Yeah.
This is for the therapy, to act as a therapy.
Correct,
the
but
designs that you have to
why
do.
should the vector price be that different?
I really don't know, but I would assume that there is a lot more in the vector design that you need to do for a therapy as compared to a vaccine. Vaccine is a one-time.
That's not a manufacturing cost, right? That's just the companies are maybe charging a lot, but it's not clear that manufacturing is that much.
No, I think the manufacturing also is the scales at which you have to manufacture, perhaps. I'm not 100% sure, but the vaccines, yes. I mean, the mRNA vaccines were not that expensive, and they used lipid nanoparticles. But the other vaccines, yes, the cost is not that much. But in this case, it could be manufacturing, it could be the design of the viral vector itself, because there are many steps that you have to involve before you actually can introduce your gene of interest into the vector, right? So could be that is what makes the cost higher.
And also, all your applications, as I saw, were with DNA.
Yes.
You haven't done mRNA.
We have. We are, I've not shown those data, but most of the carriers that work very well for plasmids, and we have done large plasmids also, like 15, 20 kb also, they also work for mRNA. So that's something that's also ongoing in the lab. Yes.
Okay. Thank you.
Yeah. So usually, like in the lab, we are comparing with standard known lipid agents, lipid-based agents. That's our standard comparison. That's how everybody does for cellular as well as for in vivo transfections. Yes. So the PBA ones, the peptide ones are not that stable. Like, you have to, the storage conditions, etc. You have to be very careful with that. But the PBAs are surprisingly quite stable. Also, they have an ester bond. You can lyophilize and keep them for months together, reconstitute and use them. The mannitol one is also, it's more stable than the other one, but yeah, with the peptide ones, the stability is not that great. The PBE ones are very good, yeah, even with the mannitol, yeah.
Hi, here. Interesting talk. I'm interested with in the PBEA the construct, and it's very curious that it's going more in the neurons and not in the other cells of the brain. I'm wondering if you checked, how large of a plasmid you can package in these,
Yes.
and do you have a range in mind?
So the one that I showed here, the mannitol modified PBA, we have done only up to 5 kb. But we now have other modified PBAs which are able to take up to 15, 17 kb.
And can still effectively cross the blood-brain barrier.
Yes.
I see. Okay. Very cool. Thanks.
Okay. So seems like there are no more questions. Let's thank Dr. Ganguly. So next speaker is Dr. Shubhadeep Ganguly. Shubhadeep is an assistant professor at the AIIMS Kolkata. He has been trained for his undergraduate, graduate, and postdoctoral training has all been in the AIIMS Delhi. Shubhadeep works. His research interests are cancer supportive care, cancer immunology, and clinical trials in oncology, with particular focus on challenges relevant to India and other low resource settings. So Shubhadeep was selected as an associate for the Indian Academy of Sciences in 2024.
So a very good afternoon. I thank the Indian Academy of Sciences for kindly extending this invitation. It has been a privilege and an honor. So I am mainly a clinician who works seeing cancer patients from the perspective of medical oncology, which means treating cancer patients with chemotherapy, targeted therapy, bone marrow transplantations, and all sorts of medications, not surgery or radiation. So when we work with cancer patients, especially in India or low middle income countries, we face a challenge, which is there is a survival gap. The survival gap means that in high income countries, the same patients are surviving at a much higher proportion compared to those we find in Indian setting. So this survival gap, we're trying to see what is that survival gap, what are the factors which affect the survival gap, and whether we can target some other supportive care while we are delivering cancer care to the patients so that we can bridge that survival gap. So what is our cancer survival in our country and all over the world for that example? So when we talk about long-term cancer survival, so no one really can say that what is a long-term survival for cancer, because cancer patients after their diagnosis can die from various causes, which can be cancer related and may not be cancer related. But in general, the long term, when we generally study patient or patient population, we say that 5-year survival, a person who is surviving at least 5 years after their cancer diagnosis, they have achieved something called the long-term survival. Even if they may die at a later point of time due to some other reasons, and even for cancer coming back, but more and less that chance comes down after 5 years. So where are we currently in long-term survival of cancer? So if we see, so I am interested both in pediatric and medical and the adult medical oncology, but cancer survival in children has been a success story, and that is what excites me a lot. So when in 1960s, when children used to be diagnosed with cancer, most of those malignancies were uniformly fatal. also for adults. But the children in the last 3 to 4 decades, their 5-year survival has significantly increased. And in high-income countries, this old data, that is 2012, it currently is at 85%, which means 100 children who are diagnosed with cancer in high-income countries today, 85% remain alive at the end of 5 years, which is a remarkable achievement. But what is that in case of adults? So this is also slightly old data, and it is a very big variable compared to the type of cancer. We know that germ cell tumor, which is the testicular tumor, it is wonderful, it is highly chemosensitive, and in today's age, in today's date, if we find a patient with a testicular cancer, more or less, they're going to survive. On the other hand, still pancreatic cancer, lung cancer, they continue to be one of the worst type of malignancies, and the survival continues to remain poor. But mind you, this is the old data, this is up to 2007. If we find this all cancers, that was reaching somewhere around 45 to 50%. And the latest data which has been come up from the SEER database of United States, they say that in today's date, even with all the adult malignancies taken together, of 100 adults diagnosed with cancer, around 60% will remain long-term survivor, which means we have also achieved a very big level of improvement even in adult malignancies, considering that adult malignancies are very difficult to treat. They're not chemosensitive enough compared to pediatric malignancies. So this is currently around 85% in children and 60% in adults. But let us come where we are in India. The same thing, same malignancy, same type of biology. In India, the survival is 55% compared to 85%. In adult cancer, it is 30% compared to 60%. So there is a 30% gap uniformly, both for children and adult. So there are multiple reasons, and I am not talking about the social reasons here. I am not talking about the healthcare system, systemic gaps. There are financial reasons, people not coming to doctors on time, cancers diagnosed at a advanced stage. But we have all the molecules. We have most of the available treatments which are available in the Western countries and here, but still there is 30% gap, and a part of it is not explained. Why it is all cannot be explained by social reasons and all cannot be explained by systemic reason. So there is a role of supportive care in this gap. And what is supportive care and how to improve it is one of the areas which I am interested in. So if we see the cancer treatment, these are all sorts of treatment which we do for cancer, but the supportive care does not include any of those. They are not directly cancer related, but they improve outcome and prevent toxicities. So what are those? So you can give some chemosensitizing agents. You can sometimes give some agents which improve their appetite, improve their nutrition. You can give some preventive vaccines. You can give some sort of prophylactic antibiotics. You can give some sort of treatment which can prevent chemotherapy toxicities so that actually you can deliver chemotherapy in a proper intensity and on a timely manner. So all of them are not directly cancer directed, but they help in delivering the cancer therapy in a much more proper way. They are applicable broadly. Most of these therapies are not specific for each and every cancer. They can be delivered to a broad variety of cancer. So targeting nutrition, it is well known that cancer treatment is known to it improves outcome. But it is known, but people really don't know how to target nutrition. It is known that a poor nutrition at the start of treatment and during treatment is bad, but it is not very well known that how better we can target nutrition. It is well known and very easy for a doctor to say to a very debilitated cancer patient that you should eat more protein, but the patient is fully cachexic, they have a very poor appetite, so they are really not heeding to those advices. So we studied a lot of nutrition in pediatric oncology. We started macronutritional and micronutritional parameters at baseline and follow up and see how it affects the outcome. We did a cohort of patients of micronutrient profile in a cohort of patients from 2012 to 2016 and then followed till 2020 and see how their baseline micronutrient profile affects the outcome. And along with micronutrient, we then also created a nutritional database from 2016 to 2021, where micronutritional parameters were measured and followed up serially, and the patients were followed up till 2023 to see how their outcome was. So this was the first thing from the micronutrient deficiency. What we did is, at the baseline, even before starting any sort of treatment, we analyzed their micronutrient parameters, vitamin B12, folate, zinc, selenium, copper, and iron, and tried to see that which are those patients who are deficient in those, which are those patients, and are those serum levels really affect their long-term outcome or not. And we actually analyzed 535 children, included over 4 years, followed them up for more than 5 years. We found that vitamin B12, zinc, iron were the most common deficiencies of children who were diagnosed with cancer at baseline. And at least 81% of patients were deficient in any micronutrient, and 14.4% were deficient in multiple micronutrients. Now, people may now say that, okay, micronutrient deficiency is there, so why don't we supplement with micronutrients? But there has always been some sort of conflict that whether to do micronutrient supplementation or not. A lot of those micronutrients have antioxidant properties, and whenever we give chemotherapy, they need reactive oxygen species to cause the DNA damage and actually kill the cancer cells. And giving micronutrient supplementation without really understanding and actually getting the reactive oxygen species down may be detrimental. We further analyzed their long-term survival, the event-free survival of the cohort and the overall survival of the cohort, and saw that it was selenium, which was, even though it was deficient in less number of patients, but selenium deficiency significantly predicted poor survival outcome in both the overall cohort and especially in hematological malignancies. So in children, hematological malignancies, leukemia and lymphoma are common malignancies. And it was predominantly the acute lymphoblastic leukemia subgroup where selenium deficiency was found to be a significantly poor survival outcome, which shows that children who are diagnosed with acute lymphoblastic leukemia, especially that subgroup, and who are deficient in selenium at baseline, even if you are giving the proper treatment, they are likely to even relapse more quickly, and they are also likely to die more quickly. On the other hand, when we analyzed solid malignancies in children, which is comparatively less common in children compared to adults, but in solid malignancy, we found that it was zinc, which was more predictor of the poor survival outcome. Here we found that zinc, rather than affecting the survival along with event-free survival, but it was more of toxic related deaths. The zinc is a mucosal barrier. It helps in the maintenance of the mucosal barrier, and we found that the children who are deficient in zinc, they're more likely to develop a lot of mucositis and a lot of toxicities due to chemotherapy, and they finally resulted into the chemotherapy could not be delivered in a proper manner, and because of the suboptimal delivery of chemotherapy, they are more likely to have a toxic related death, and then also more likely to have relapse. Then further we tried to develop a prospective nutritional database of children. Around 2,000 children were recruited, and they have been followed up from baseline and regularly at 3 monthly interval while undergoing cancer treatment. We measured their height, weight, mid-upper arm circumference. The height, weight and mid-upper arm circumference, everything measures the separate things. The height in children is a marker of chronic undernutrition. We measure a parameter called as height for age, and a person who has stunted for age is a marker that they has been chronically undernourished for a long period of time, even much before the diagnosis of their cancer. On the other hand, weight for height, which is a parameter which is a marker of acute malnutrition, where there has been a sudden decrease in weight, even if they have actually achieved a proper height, that may be affected by their diagnosis of cancer and poor appetite during the malignancy. And mid-upper arm circumference is just a surrogate marker of their muscle mass. So we measured all these parameters, and when we measured these parameters longitudinally, and we categorize them that gradually, when they have been followed for more than 5 to even up to 7 years, a few patients, there have been 3 groups of patients. So patients who gained more than 5% of their weight after starting their cancer treatment. There have been patients who continues to remain plus or minus 5% of their weight. And there have been few patients who has a loss of 5% of their weight. We consistently found both in event-free and overall survival that this is the group where even with the start of cancer treatment, the patient unable to gain weight. In any normal children, they gain weight continuously. During cancer treatment, when the cancer treatment starts taking effect, the cancer cachexia goes down, their appetite improves, and they should actually gain weight much more rapidly. But there will be a subset of patients who will continue to lose weight of more than 5% is a specific cutoff which we chose. But the idea is that children who are unable to gain weight, even undergoing cancer treatment, they will consistently have a poor survival outcome. And the baseline we found that it is the chronic undernutrition. Those who have a height for age less than -2 standard deviation at baseline, which is a definition or cutoff which we use for chronic undernutrition, also predicted poor survival outcome. So we have all this thing and we analyzed, okay, yes, we understand the chronic undernutrition is bad. We understand the weight loss is bad. And we also found those things that this is also well known in adults that cancer cachexia, someone who, at the diagnosis of cancer who has a weight loss of more than 10% within the previous 3 months during their cancer diagnosis, they're likely to very much die within 6 months. So those information we already had. So what to do? So we conducted a trial of olanzapine. So olanzapine, as we know, that it is a atypical antipsychotic. It is given to patients with schizophrenia. It acts on dopaminergic receptors. And along with dopaminergic receptors, it also acts on histaminic and serotonergic receptors in the brain. So it is a drug which is used by psychiatrists. But it is an excellent antiemetic. So we kind of repurposed it as medical oncologists. While treating patients with chemotherapy, we used to give them short course of olanzapine, and it is an excellent antiemetic and used to prevent chemotherapy-induced nausea and vomiting. It has been used in adults to see how in adult patients, it has been shown that it improves weight and it also improves appetite. And adult patients with cancer cachexia, they develop, after giving 3 months of olanzapine, they seriously have a very significant proportion of patients develop weight gain of more than 5%. So we tried to see whether this holds true in children as well. So we did a multi-center phase 3 trial in 3 centers in AIIMS New Delhi, in JIPMER, as well as Adyar Cancer Institute in Chennai, where 130 children with solid organ malignancies were randomized to olanzapine diet versus only diet arm while undergoing cancer treatment. 3 months of olanzapine was given to evaluate whether proportion of children with more than 5% weight gain improves on addition of olanzapine. Surprisingly, we did not find there was any change in the 2 arms. Both the arms, 39% versus 35%, addition of olanzapine did not help in improvement of weight gain in children. This is in contrast to adults, where adult it has been shown benefit and is currently now recommended also as a part of society guidelines for patients with cancer cachexia to prevent weight loss. So we are currently studying, there are various reasons for it, because the type of biology in adult malignancies and the pediatric malignancies are slightly different. The adult malignancies, the pro-inflammatory profile, the cancer cachexia is more dominant, while in children they're very chemosensitive, and there is a lot of malignancies in children where steroids are given commonly, and so steroids perhaps have blunted the effect of olanzapine. So trying to see the plasma biomarkers currently which predicts the weight gain during cancer treatment. So overall, we observed that chronic undernutrition at baseline, micronutrient deficiency, and weight loss, these 3 factors adversely affect survival outcome in children with cancer, but interventions to reverse those are currently limited, and that is what we are currently working on. I thank All India Institute of Medical Science, New Delhi, where I worked on all these studies, and currently AIIMS Kolani, where I'm currently working. And I thank you all for your kind attention.
So maybe I'll start with a question. So Shubhadev, you did not mention that although there was no increase in weight, the proportion of children for whom the weight decreased, that was different, right? When you
No, that was not different. So we just calculated the proportion of greater than 5% weight gain. So that also remained same, and the person who the weight remained within plus or minus 5% or any weight loss, that also remained same. So the three categories were broadly similar in the two arms. So yeah, so I have not, so this is just two groups. So who gained weight and who?
No, I'm looking at the figure on the right.
This one, right?
Yes.
Yeah, so this is the decreased in the weight.
It is 25 versus 33.
Yeah, so that is not significant. There was no statistical.
Did not come out to
No,
significant. Okay. The other question is, why is the trend changing in bladder cancer? So in your first slide, every cancer, the survival actually improved except bladder. Why is that so?
Which ones?
The first slide.
This one?
No. First one. The overall epidemiology.
Yeah. So yeah, that is a completely different thing. So initially, that is an epidemiological problem. So initially, when non-invasive bladder cancer was, there is two types of bladder cancer, which is muscle-invasive bladder cancer and non-muscle invasive bladder cancer. So initially, when the epidemiological calculations were being done, the non-invasive bladder cancer were also being included.
So the definition
has changed. Yeah.
Okay.
So
all right.
Yeah, please.
So, Shubhadev, one quick question. Very nice talk. One quick question with respect to the long-term survival.
Yeah.
You're only looking at these factors that are intrinsic to the individual, but you're not looking at the exact mutations. Is it possible? I'm just loud thinking that those who are not long-term survivors, they have some specific bunch of mutations, while those who are surviving more, you didn't relate them to the mutations. That's one question. And the other simple question is, you showed a graph where for prostate cancer, it was completely different compared to the other organs, and why?
Yeah. So coming to the first question, it's actually a wonderful question. So all malignancies, there is some genetic component which predicts their survival. So while analyzing, we have adjusted for the risk groups of the malignancies, and whatever the analysis and the hazard ratios, those are adjusted for the risk groups and the staging. So which are the known confounders of the survival outcome. Coming to this study, whatever, so where is the prostate? I have not.
The prostate is the one which is sharpest, right?
Yeah, so I think that is probably when we started using androgen deprivation therapy. So androgen deprivation therapy.
Let me try and give you another explanation.
Yeah, please.
What has happened is the way prostate cancer is diagnosed has changed.
Yeah, that is another.
These are all prostate cancers. Now we diagnose them by doing PSA and you pick up. And you know how prostate cancer is, whether it's a cancer is a question. And that's where my question was, why I raised my hand. When we compare 5-year survival in cancers in adults in the West versus in India, the nature of cancers may be very different. Prostate cancer forms about 25% of the cancer in the West. And many other cancers there are being diagnosed by screening. And you would have a lead time bias and those biases. Whereas in India, we are picking up cancers by after symptoms occur, and also they are picked up at a late stage. So that's part of the thing. Secondly, I'm not actually surprised that you found micronutrient differences and that those who lost weight did poorly compared to the very good talk. But, the problem is that there is in medicine and, there is a bias of, reverse causation. Those who have more advanced disease are the ones who are likely to lose weight compared to those, So basically the weight loss and weight gain separates those who have more aggressive disease versus less aggressive disease. And that's part of the reason. So it's a cause rather than the consequence, and therefore treating it may not make a difference.
Yeah. So we actually don't know which one really came before, but in a way we have adjusted for the stage and risk as I explained. So some of the risk factors which affects the weight loss and other things is taken care by that adjustment by the analysis. But this part is known that patients with cancer cachexia, if we target their weight loss by giving supportive and adjunctive therapy, we improve their survival along with the cancer treatment, whatever else we are giving. And so olanzapine, the survival also improved in adults by giving additional olanzapine. There is a new molecule which has come up, which is ponsegromab, which affects GDF15, which is a cachexia causing molecule. So that also has shown that in cancer that it improves the survival. So targeting weight loss during cancer treatment is not something that we should ignore. We should also treat it separately.
Yeah, and the micronutrient fortification that you have done, the point of bioavailability and bioaccessibility has to be differentiated. If that is not differentiated, then the confusion will arise. You may give, but in one case it is bioavailable, in the other case it is not bioavailable. In one case it is bioaccessible, the other case is not bioaccessible. So it's very important, just by giving it a dose, we assume that we have made a control experiment. But actually the individual response of bioavailability, bioaccessibility, and also assimilability, bioassimilability has to be differentiated. Otherwise, the cohort will be wrong decision or wrong data that shows up. Thank you.
Yeah. So I just wanted to add to the comment. So we have not done any micronutrient supplementation experiment. One of the reason I told that there is some concerns that micronutrient may affect the chemotoxicity. So we have not done that. Another thing we have not done that this micronutrient deficiency, it may be a reflection of the inflammatory state in the body. So in inflammatory state, the micronutrient level goes down, and so their level has to be adjusted based on the blood CRP levels and all those things. So actual level of micronutrient, whether it is deficiency because of their dietary thing or because of increased usage because of their inflammatory state, that part we have not differentiated. So that kind of answers your question.
Excuse me, sorry.
Yeah,
it might sound like a little naive, but my question is that, like you said, you gave a certain drug to in adults, it was seen that their appetite was increased and it was helping them in the survival, right? What if, let's say, the person you were giving the drug to, their oncogenes develop a resistance to the drug, and if the person gets cancer second time after getting treated, do you think that if you give the drug again and it has resistance to it, so is there any and they lose weight more than they would have lost in the first time, so is there something to avoid that or something?
Yeah, so this drug does not act on the cancer cells, right? So it is a kind of an antipsychotic which acts in the brain. Okay, so it is not acting on the cancer cell, it is acting on the appetite regulation pathway of the brain. So there is no question of having resistance, the cancer developing resistance to that drug, right? Yeah. Okay, thank you.
Okay, so the last speaker of this session is Professor Satish Raghavan. Satish also works in cancers, primarily liquid cancers. Satish did his postdoctoral research in the University of Southern California, United States, and he is an investigator at the IASC. He got the Shanti Swarup Bhatnagar Prize in 2013, the Leukemia Research Foundation Award in 2010, the Nasir Reliance Industrial Platinum Jubilee Award in 2015, the S.P. Rai Chaudhary Lecture Award in 2023, the Society of Biological Chemistry Ram Rao Lecture Award in 2024, and J.C. Bose National Fellowship 2024. So Dr. Raghavan is a fellow of the Indian Academy of Sciences, the National Academy of Sciences, and the Indian National Science Academy. And he was elected a fellow in 2016.
Thank you. I hope you are able to hear me. Thank you, Chair. And also thank you, Indian Academy of Science, for giving me the opportunity to present here. Today I'll talk about the molecular basis of chromosomal translocation. We just heard the talk about hematopoietic malignancies and other cancer. But here I'll talk a little bit about the molecular mechanism associated with the generation of translocation, particularly focusing on DNA conformation and the role of immune machinery. Before I start, I'll just give a brief introduction of the kind of research what is being done in my lab, which are all centered around DNA double-strand breaks. It is one of the most lethal type of DNA damage normally occur in our cell. Now for the audience, if you are not familiar about a double-strand break, this is nothing but two neighboring breaks in both the strands of the DNA, as shown here, the close vicinity. Now, normally, whenever this kind of breaks occur in our genome, the continuity of the genome is affected. So therefore, there no replication, transcription, and which can lead to many type of issues, including cancer, what we just heard. So, but whenever such break occurs in our normal cells, we have an efficient DNA break repair system, DNA double-strand break repair system, as shown here, which ensures the integrity of the genome, which means our cells are happy and we are happy. But in cases where when misrepair of the DNA ends or the chromosomal end happens, and that is where chromosomal rearrangements and the translocations and such kind of abnormalities takes place, and it could lead to various diseases, including cancer. There are cases when there is no repair takes place in the nucleus, right? So that's what lead to cell death and then different mechanisms. And so my lab essentially work on all these aspects, but today I'll be focusing on more of chromosomal abnormalities associated with hematological cancers. So now when you look at the distribution of chromosomal translocation, mostly about 80–90% of leukemia and lymphoma are characterized by one or other type of chromosomal translocations. And going back to the history, somewhere in 1960s, that is the first time such translocations were discovered, just by using cytogenetics methods. Janet Rowley first described the Philadelphia chromosome, which is seen in chronic myelogenous leukemia. Then there were array of discoveries in next 20 years describing several translocations. And then early mid of 80s, that's where people started trying to look at why these chromosomes breaks and what is so special about it. So what they were trying to do was they were trying to clone and sequence, that's the era molecular biology started booming. And there were so many information generated in, say, next 10–15 years, sequencing several different translocation breakpoint junctions. Somewhere late 2000, that's where people like us started working on what is the meaning of all this information generated, why chromosomes break in certain people or certain genes, and then that gives some sort of selective advantage to those cells, which helps the those cells to transform and then become cancerous later with an array of additional mutations. So just to, again, make it clear for non-biology audience, so these are two chromosomes, and whenever there is a translocation, essentially translocation is nothing but exchange of chromosomal arms. But the primary requisite for generation of such translocation is you need to have chromosomal break. Chromosomal break essentially means breakage in the genome, double-stranded DNA, right? And that's what is shown. This is a translocation between chromosome 14–18, which is normally seen in follicular lymphoma. Long time back, it's about, I think now it's about more than 20 years back, we have come up with an important finding where we showed that the break, in this case, which is T1418 translocation seen in follicular lymphoma, occur in BCL2. Of course, that was not our finding. But what we found out was there is something called major breakpoint region in BCL2, and it could fold into something called non-BDNA structures. I'm sure the audience here knows what is a BDNA, which is Watson-Crick hydrogen bonded double-strand break DNA, right? So what we found out here was the DNA at this region, not in B form. We showed several evidence to believe that there is a single-stranded character. And then we also went on to show that there is a protein called RAG, recombination activating gene. These are normally responsible for immune system diversification. I'm sure students will know, right? And this protein can aberrantly cleave these kind of structures. That's what we showed during my postdoctoral studies. Then later when we started working here in IISc, that was one of the first questions asked, what is this non-BDNA structure we were talking about? Now, when you look at the literature, these are examples of several type of non-BDNA structures. So these are formed, for example, cruciform DNA, triplex DNA, hairpin, tetraplex, aDNA and zDNA. It's there in textbook, well accepted one. And they all form when there are specific sequences, repeat sequences present in our genome. I'll not get into the details because time is limited. So essentially you need to have those specific sequence to have such, special or secondary forms of DNA formed in our genome. And honestly, these structures were all described more or less same time, 53, 54, when original BDNA were discovered. But they never got enough importance, or honestly, people did not believe that this will have some sort of biological function. Mridula, one of my first PhD students, showed that the structure formed during T14 translocation is something called G quadruplexes. These are the structures formed when there are stretches of guanines present in the genome. Then it can fold into some sort of a quadruplet structure as shown here. And based on that, we come up with a model wherein we showed chromosome 18, that's where the BCL2 gene is present, break based on the DNA structure, but it's cleaved by RAG complex, which, as I said, is important for VDJ recombination during immunoglobulin diversification, and which is a sequence-dependent process, whereas this is a DNA structure-dependent process. And when we try to look at several translocation junctions, information was available already in the database. We found out that not only in T1418 translocation, BCL2 major breakpoint region, even other translocation breakpoint regions from the patients actually have these sequence motifs that can support formation of such secondary structures. And so that's where this is what one of the aspects today I want to, maybe speak next 3, 4, 5 minutes, where we try to understand how BCR-ABL, these are the two genes broken during formation of Philadelphia chromosome. As I said, the one which was described in 60s, 70s. And then if you look at the patient breakpoint region, you see there are a lot of arrows here that indicate a particular patient breakpoint region. There are a lot of G quadruplex forming motifs, so which is again shown here. And what we did first was we tried to do a particular experiment, which is something called sodium bisulfate modification assay. I'm sure you would have heard about this name, sodium bisulfate modification, normally used for studying DNA methylation. So you unwind the DNA and then treat with the chemical, and the principle is nothing but sulfonation of cytidine when it is present in single-stranded DNA in acidic pH, and then so that the cytosine can get converted as a uridine. Right? And then following PCR and sequencing, you will read this as a C to T change, cytosine to thymine change, which is a easy way to find out originally in the genome whether the DNA region was single-stranded. So now what we found out was in BCR region, there are many stretches of single-stranded area. And then we went back to the in vitro biochemical assay systems where we designed several oligomers from those G quadruplex forming motifs. As I said before, these are supported by stretches of guanines. So the complementary control region will be cytosines. Now, if you run on these kind of oligomeric DNA radio labeled on a native page, What you see here is, I mean, by the way, the structure formation here is dependent on potassium chloride. So in presence of potassium, you can see, I mean, one of the example I'll show you, the guanine started moving much faster compared to minus potassium gel, which gives an indication of, oh, there is something happening there. But then, of course, you do, mutation of each one of those structures to demonstrate that they are indeed something called intramolecular G quadruplexes. Then there is something, I'm sure you all heard about, circular dichroism, which can be used for studying structural variation of DNA and protein, right? So we could, I mean, again, I'm not going to get into those details. And then DMS or dimethyl sulfoxide protection assay can be used to, understand the base pairing in this structure. And this is a 2D model there. And now the issue here is, this is all oligos in vitro. People will ask, what is the relevance? It's all fine in a tube, but what is the evidence that it will form within the cells? But then before getting into that, so here we cloned this region into a plasmid, which is like, double-stranded circular DNA. I'm sure non-biologists also will know that. This is a simple assay system, something called a primer extension. So denature and then radio label the oligos. And then if there's a structure formed, G quadruplexes formed, then during the extension, you find a post site. Whereas in the complementary region of the DNA, where you will have cytosins, of course, no G quadruplex formation, then you don't find the similar kind of a pose. Now, if you mutate the G quadruplex forming motif, then you can see it's disappeared. And then if you change the positions of primers, you can also see the pose sites moving. So this is what we try to see within the cells whether this region can break. So what we did here is we used episomal constructs, and it's like a plasmid, but it can replicate. In this case, it can replicate in human cells, mammalian cells. Again, to make a long story short, we are able to replicate the translocation breakpoint region using this episomal DNA. And then this red sequence, what is shown is what is deleted. And these are all clones who are sequenced, confirmed, and all those things. And then we went on to show that there is an enzyme called AID, activation-induced cytidine deaminase. This is a deaminase, and which can bind to these structure regions, as shown here. And then it can deaminate the cytosins and leading to the mismatch, and then finally, I'll show a model there. This was also replicated by using purified proteins. So this is our working model. So there are multiple G quadruplexes are formed at the BCR region, and then the AID deaminate the cytosins, and then there is something called AP endonucleases, which can cleave at the the mismatches, and failed base excision can lead to DNA break and translocations. And next 2-3 slides, I'll just summarize. This was another study where BCL6 breakpoint region seen in diffuse large B-cell lymphoma. Again, the patient breakpoint can form different types of structure, including cruciform DNA, triplex DNA, and then there are different clusters where even G quadruplex itself could lead to fragility. This was another example where we showed that RNA DNA hybrid formation or R-loop formation could result into, could make the MYC region fragile in Burkitt lymphoma. So to summarize this portion of the work, essentially our study shows that non-BDNA structures are formed at chromosomal translocation breakpoint region in lymphoid cancers. In presence of G quadruplex DNA, triplex DNA, cruciform DNA, R-loop structures contribute to the fragility at the breakpoint regions. DNA replication and transcription, then DNA supercoiling facilitate the formation of such structures within the genome. And enzymes like RAGs and AID can act on non-BDNA, leading to chromosomal breaks. Maybe another 3-4 minutes. I'll just try to talk a little bit about the this new property what we discovered about the RAG as well as AID. So this is what happens during normally during BDJ recombination in our B and D cells to ensure the immune diversity. So this enzyme, RAG1 and RAG2, it acts as a dimeric complex, binds specifically to recombination signal sequences. These are conserved sequences. That's why this process is also called site-specific recombination, because it just specifically cleave up the 5' end of the heptamer. Then there is also a monomer in both these cases. So now what we did was we tried to do some biochemical assay system by using purified rags from human. This is the purified protein, and then we use the physiological substrate, which is the VDJ recombination facilitating substrate, and the other one is the non-BDNA structure where bubbles are used. So Abni went on to show that when you incubate with the purified protein, actually even this case, when rags act as a structure-specific nuclease, there is a sequence specificity. For example, when cytosins are present, both top and bottom strand can be cleaved exactly at the double-strand, single-stranded DNA junctions. Whereas when thymines are present, it will cleave. The efficacy is much poor, but when purines are present, this protein cannot touch. Rupa went on to show that not only the sequence of the single-stranded region, even the flanking double-stranded sequence matters. And there are particular domains. There is something called a non-BDNA binding region in RAG1. If that is important for physiological function, whereas the ZN-C2 module contribute towards the structure-specific nuclease activity. Nishana, so she did a very interesting, simple experiment. What she did was she placed the monomer of VDJ recombination next to these kind of structures. Okay, so what she found out was now all these DNA structures, which was otherwise not sensitive to RAG, become sensitive because you can understand because RAGs bind to the monomer, then cleaves at the single-stranded, double-stranded DNA junction. And she also showed even if you have a single nucleotide mismatch, that is good enough for good enough as long as there is a monomer is present. Now this was a Cell paper published sometime 2008 from my previous lab, where they found that CPGs are clustered near patient breakpoint region, and if there is a deamination of methylated cytosine, it could lead to TG mismatch, which causing the translocation breakpoint region. So at this point of time, we ask the question, how often such monomers, what we described before, may be present in the translocation breakpoint junctions in patient sequences. So we analyzed more than 3,000 patient sequences and found that about 93% of the cases, you have monomers present at the patient breakpoint junctions. And of that, 74% of the cases, you have a CPG right next to that. So these are a lot of examples I'll not get into. So now by using biochemical assays, we showed that purified RAGs can bind to those fragile regions. Not only bind, it can cleave. We also have done chip assays to demonstrate that RAGs indeed bind in those regions inside the cell. And this was LM-PCR or ligation-mediated PCR to show that these breakpoints seen within the cells are comparable to what is seen in the patients. Based on that, we come up with the model wherein RAGs bind to the mismatches created because of the deamination of CPGs. And then bind to the monomer and then cleave at the mismatch at the CPG site. This is where I am going to stop. Essentially, we found additional layer of regulation of RAGs in mammalian cells. We identified two microRNAs. One is MAR29C and the other one is 29A. Both could regulate the expression of RAGs in a B cell, T cell stage dependent manner. I think this I'll skip. And to summarize this portion, RAG can act as a structure-specific nuclease, cut non-B forms of DNA leading to chromosomal breaks. Additional mechanisms such as presence of cryptic signal sequence and CPG lead to translocation. Tight regulation of the formation of DNA structures by resolvases and expression of RAGs aid is important. MicroRNA 29C, 29A provide an additional layer of regulation during RAG expression. And this I did not show. RAGs are expressed in glioblastoma cells and could facilitate the formation of chromosomal rearrangement. This is something interesting because generally people believe this protein is expressed only in B and T cells, and we are working more on that. These are some of my collaborators, and then funding from all government agencies, and also some private agencies. I'm really thankful. And this is my current group. Then, of course, a lot of old members also contributed to the work what I presented today. Thank you.
Satish, right here, right in front of you. Yeah, very interesting talk. I was just simple question. I mean, how frequent are these non-number-like sequences in normal humans?
Yeah. No, this is something reviewers made us to evaluate. When we did, it's about 3 to 4-fold more than, less than what we have seen in the patient breakpoint region. Yeah, so this is something we checked. Okay. Okay, thank you.
So let's thank all our speakers for today. I mean, that was the last talk for this session. Let's thank all our speakers for today for their wonderful talks. And we break for tea, right? Okay. All right.
We break for tea now, and at 5:30 we will have a public lecture which has been announced in the program, and so please do come back for that. Yeah.